Hey, in this video, we are going to look at the organ for the water and mineral salts uptake in the plants. So actually, this organ is referring to the root. Okay, and this answer the questions of why the root is the most important organ for plants. As you have learned in chapter 1, roots are involved directly in plant growth. And you know the location of the roots, usually it is located underneath or under the soil. And this is because the root grows towards the center of the earth and also watery areas. I wonder if you still remember the terms geotropism positive and also hydrotropism positive. So roots are showing this type of response to gravity and also to water, and that's why we call it as geotropism positive and hydrotropism positive. And hence this explains, or this helps us to remember or to know the main functions of the roots, and that is to provide support and strength to anchor the plant in the soil and also to absorb water and mineral salts from soil and to transport them to the stem and leaves through the xylem tissues. This is the root structure of the root structure, the longitudinal section, where you can easily identify these extended epidermal tissues, and that is the root hair. Okay, you have the lateral root. Okay, the lateral root, which is not the primary root, and also the cap down here. This is called the root cap. Also, you have the root collar, which is very near the surface of the soil. Okay, let's continue to look at the functions of the structures mentioned. The root collar is actually the part of the root that is connected to the base of the plant stem. Okay, root hairs is actually the modified epidermal cells. Okay, so this is the epidermal cells. Okay, the outer layer here, which serves as protection. Okay, and then this root hair emerges from it. So root hair is the modified epidermal cells. And why does this root hair need to be in this extended manner, elongated manner? It's because the function of the root itself is to absorb water and mineral salts, and the plants need to absorb a lot of them. So to increase the uptake of water and mineral salts, they need a total surface area, a big total surface area. So by having extended manner like this, okay, the surface area of the roots can be increased. The total surface area can be increased. And that is the reason why. Okay, this is the root cap down here at the tip of the root. It is meant for protection, just like when you're wearing a cap. Okay, you want to protect your head from the direct exposure of sunlight. So same goes to the plant over here, the root. Okay, it is the surrounding, you see the surrounding, the environment is the soil. So when the root penetrates deep down into the soil, it has to protect itself from damage because the environment that it is encountering is not friendly. It is the harsh environment, and therefore the cap is there for protection. Okay, next is the internal structure of the roots. So very easy to identify by looking at the extended epidermis over here, or the modified epidermis, and that is the root hair. Okay, just right beneath it, the one just now I've mentioned, that is the epidermis. Okay, this layer over here, okay, this is the cortex. Okay, now we trace here to this, the first ring, white color, the endodermis. right underneath the endodermis, we have the pericycle. Pericycle inside here. Okay, this middle portion is the primary xylem, and you have the primary phloem. It's like the star shape, or the, if you are to look at it, it's like an X for me. Okay, X stands for xylem. So whenever you see X like this in the middle, okay, immediately it tells you that this is the structure of a eudicote roots. Eudicote roots, not monocot roots. Okay, this vascular cylinder, okay, is it is consists of the primary xylem, pericycle, and the primary phloem. Okay, made up of these, the vascular cylinder. Okay, encapsulated over here. So epidermis, the gray color region here at the outer. These cells are closely arranged. Their cell walls are thin. They are permeable to water. Okay, as permeable to water, water permeable to facilitate water movement into the roots. Okay, the modified epidermal cells forms the root hair cells. So elongating towards lateral. Okay, and we already see the significance just now to increase total surface area, increase water uptake, and also mineral salts uptake. Okay, then the root hair cells are not layered with cuticle. So in other words, cuticle, this is the waxy layer. Still remember that it is insoluble. It doesn't allow water to penetrate through. But in this case, root hair doesn't have this cuticle, and therefore it is able to allow water to pass through, allow water absorption. Also, these root hair cells, they have big vacuoles. Okay, the reason is to store more water and mineral salts, and therefore increases the capacity of the root hairs to serve in water absorption. Okay, cortex. Okay, this region over here is the cortex. Okay, consists of thin cell walls which also facilitate water movement in the roots. Just located just underneath the epidermal layer. You see, just next to it. They are loosely arranged as well to facilitate gaseous exchange. And mostly they consist of the parenchymal cells. Okay, I wonder if you still remember the ground tissues, the parenchyma, sclerenchyma, and the collenchymal cells. So cortex here mainly they are formed by the parenchymal cells. Okay, so let's continue on to look at the endodermis. Now this is the third layer. Now just now we have the epidermis, cortex, and the endodermis. It's the one that is actually separating the cortex. Okay, you see, cortex and also the vascular cylinder. Okay, so endodermis is separating the cortex from the whole thing, entire thing here, the vascular cylinder. Okay, we're talking about endodermis. So most of the endodermal cells, they have this structure, the suberin or the lignin thickening at the walls. Okay, and this thickening, this structure is known as the Casparian strips. Okay, they form the Casparian strips. Okay, so what is actually Casparian strips? It is water impermeable sealing. Okay, that's like a seal that fills the space between the cells of the root as well as the root epidermis. Fills the space between the root between the cells of the root epidermis. Okay, Casparian strips. Casparian strip. Okay, so that means since they are water impermeable, so water, how is water going in? Since you know roots function is to absorb water and mineral salts. But then we say that this Casparian strip is water impermeable. So how is water entering diffusing from outside here, surrounding soil water into the xylem. So it is through the cytoplasm, it is through the plasmodesmata. So endodermis is the one, the cell itself is the one allowing water and mineral salts to be absorbed, to enter into the vascular cylinder. Okay, not air bubbles. So that means, okay, I show you another picture here. Okay, the pathway. So you get to see just now, this orange structure, that is the Casparian strip. Okay, this is the xylem which transports water and mineral salts. Okay, this is the pericycle just right beneath the endodermis, right? So we have water going from this direction, epidermis entering the cortex, subsequently into the epidermis, pericycle, and into the xylem. Then water is transported from the roots to other parts of the plants. So this pathway is the simplest pathway. Okay, it's the simplest pathway like this. Casparian strip, just now we say it's water impermeable. Therefore, the significance is that it will prevent backflow of the salt solution from the xylem, okay, back to the cortex. From the xylem over here, back to the cortex, this cortex region. So because having this Casparian strip, there's no backflow of water lah, and then it blocks passage of substances through the pits of the cell wall, and therefore substances have to move into the cells of the epidermis. So in other words, must be within the cells, the movement. You see, it is within the cells here. So it doesn't go this way, but then it must be here into the cell itself, the endodermis. The cells of the endodermis. So the movement of water and water-soluble substances can be controlled by the endodermis cells. Okay, so it is through this pathway, the endodermis cells. So this is like no leaking back like this because of having these Casparian strips. Okay, vascular cylinder. What is the vascular cylinder? It's the root core consisting of the phloem and xylem tissue. Okay, surrounded by one thick pericycle cell tissue. This is pericycle. So that means, okay, go back to the structure. This is now a bigger one. Vascular cylinder here inside has got the xylem, has got the phloem. Okay, that's the vascular cylinder, and then the pericycle is here, encapsulating it. So here, surrounded by the pericycle just now. Okay, surrounding xylem and phloem, that is vascular cylinder. So pericycle is involved in secondary growth and also formation of the lateral roots. Okay, still remember secondary growth. So that means there will be formation of secondary xylem, secondary phloem. So usually in this eudicote cross-section of the roots, the xylem and the phloem, they are arranged in a star-shaped pattern. This is called the star-shaped pattern. Okay, and the xylem tissues transports water and mineral salts. Okay, phloem tissues transports organic substances, sucrose, plant hormones. So again, this is a revision for you. Okay, for you to recall the function of xylem tissue and phloem tissue. Okay, we're talking about the vascular cylinder. Okay, let's move on. Okay, like right, this experiment, it says to observe prepared slides of cross sections of the monocots and the eudicots roots. So for zymase corn plants, this is represented by the eudicots. Okay, representing eudicots. You have the, you see, look at the discussion. Okay, draw the cross section of the Zymase and Tilia species roots. Record the magnification. So, sorry, just now I mentioned the corn plant. That is the monocot structure. Monocot plants. Okay, so discussion, let's look at the questions. It says, what tissues form the cortex? Okay, just now we already see over here, the cortex is mainly consists of the parenchymal cells. So we can put here our answers straight directly. We can write down parenchymal tissues are the main ones that forms the cortex. Okay, how can these tissues be identified? The parenchymal cells, they have thin cell walls. Okay, you get to see the thin cell walls unlike the sclerenchyma and collenchyma, and the vocals are big, okay, for water storage. So for monocot roots, okay, the second question is, how are xylem and phloem tissues arranged in roots of monocot and eudicot? So you can see this schematic diagram and figure 3.5 over here. They are the structures of the eudicot roots. So you get to see the star shape in the middle. So it says for eudicots, the xylem is in the middle of the root. The phloem is outside the xylem. This middle and then outside. Phloem, middle, xylem. Okay, for monocots, there's another picture over here. You can see that for monocots, the xylem phloem, they are arranged in alternate ring manner. You get to see the xylem and then the phloem. So in alternate ring. Okay, question number 3. Based on your knowledge in biology, why is the arrangement of xylem and phloem tissues different in monocots and eudicot roots? Why is it different? So we can use the point of usually the eudicot roots are the ones that undergo secondary growth. So that means they have the vascular cambium. Okay, you still remember vascular cambium, the tissues that promote secondary growth. So that means from vascular cambiums, the secondary phloem, secondary xylem will generate from it. So this, the cells over here, they divide through mitosis, forming secondary xylem and phloem. Okay, but for monocot plants, usually most of the monocot plants, they do not have this vascular cambium. Okay, that is about it on the discussion for 3.2, the root being the organs for water and mineral salts uptake.